Okay, so today's video is just going to kind of cover what to expect for Lab 7. And in this lab, we're going to be covering photosynthesis and respiration in plants. Obviously, this is fairly different than human or animal cells that only have mitochondria. Because in plant cells, they also have structures called chloroplasts that can carry out photosynthesis, essentially making chemical energy, such as glucose, from energy that they harvest from the sun. So within a single chloroplast, two main functional systems are being carried out, and that's Photosystem 1 and Photosystem 2. This is a lot like the electron transport chain, at least over here, that we encounter in animal cells, with some small differences. For the purposes of the lab, you probably won't need to know much about that. You can review it for class though. So the first thing that comes through where we get some of the electrons that we're going to need to load into this chain over here are from H2O. Not only that, we use light energy, so light has to come in, and it can excite electrons from here and kind of steal them off this H2O, and the byproduct of that is O2. Here, we can just get rid of this. So that's a basic overview of Photosystem 1. From Photosystem 1, you get two products. One is reduced NADPH, and this is reduced because it's gained an electron, this form has. And you also produce a little bit of ATP. Photosystem 2 still relies on water, but it mainly relies on CO2 coming in. Now, CO2 is the source of carbon from which photosynthesis can build chemical molecules. And so, only recycles, but it can make glucose, which can become starch, cellulose, things like that. And it takes away the ATP and leaves us with ADP. And it also leaves us with the oxidized form of NADP+. So in this case, this has been oxidized, and this actually feeds into this cycle over here. NAD+ takes an electron from H2O, excited by light, combines into NADPH. The cycle can repeat. So for our purposes in lab, the way we're going to measure this cycle, it's similar to how we measured the Mallet reaction, which is an animal cell mitochondrial reaction, I should say. But in this case, we're going to have something called DPIP, and it's a small dye that when we insert it into the cell at first, it's blue. And it's oxidized, it needs to take an electron, and it's blue in this state. So if this isn't happening, this photosystem 1, if we have no light, for example, will just stay blue here. Because if you oxidize this, it'll turn into DPIPH, yep, DPIPH, and this is colorless. Okay, so DPIP is the molecule that we're going to replace NADH with, so to carry the electron. And we can measure it in the spectrophotometer because if this reaction isn't going on, we're just going to have a lot of blue oxidized DPIP. If this reaction is going on, we're going to have a lot of DIPH or DPIPH, and it'll be colorless. So we'll see a reduction in absorbance. So if we take a look at this, if we have absorbance over time, in this lab, you're going to be asked to have 3 cuvettes. One is going to have fresh chloroplasts in it. Another is going to have, another's going to have fresh chloroplasts. The other is going to have boiled chloroplasts. So these have been kind of messed up. So nothing should happen in this one. And this is your fresh dark and fresh light. So we have boiled versus fresh chloroplasts. That's that difference. The next one is that the dark reaction, we're actually going to keep in a little foil case during the whole experiment, so it never receives any light. It's never going to receive any light energy to do photosynthesis. So what we should see is a lot of reactions and a fast reaction from the fresh light reaction. So if I was to document this, we would start out with our DPIP up here, and it should go down. So say this is fresh light right here. It should go down very quickly, and it should all be oxidized to the colorless DPIPH. Oh yeah, that's colorless. So we start with a high absorbance, but that gets oxidized by the chloroplasts if the cycle's happening. However, if we are in the dark or our chloroplasts don't work, we start with DPIP, and we essentially, some might happen, some oxidation of this, but essentially you end with DPIP. And a reaction like this that's really slow equals no photosynthesis, because there's no light or the chloroplasts just don't work. So it essentially couples these two things as the same idea. Your chloroplasts won't work if there's no light. The next one, so that was kind of examining photosystem 1. Remember from the beginning of the video, that's where light needs to happen. The next reaction is the one in photosystem 2, where CO2 needs to come in and ultimately leave as glucose. So we are actually we have little apparatuses that can measure CO2 just as the gas. We don't need to do anything with spectrophotometer, and the computer is going to tell us how much of this CO2 is going to be consumed by two different reactions, one in the light and one in the dark. So in this case, our light reaction, our CO2, we're going to be able to view if our CO2 is going to drop. Here, let's say this is light. And we'll also be able to see that if in the dark, if the CO2 stays around longer. So this is just a measure of CO2 over time. So remember, a reaction like this that's sharp, it happens in a small amount of time. And the CO2 in this case, say this is the dark reaction, it won't occur. However, since photosystem 2 can occur in the dark, it would, we'll be able to measure this and see, we could see something like that, the CO2 could actually still be metabolized. So I'm actually excited to see about that. Okay, and that should do it for this week's review.